Okay, genomics. So genomics, put simply, is the study of our genome. And in the next 15 minutes or so, I'm going to tell you what our genome is and what it is that we study. Now, most of you, I hope, but many of you will know that our bodies are made up of cells, and they're made up of trillions of cells, like more than you can imagine. And the majority of cells in our body, let me just get the pointer. So this is a cell. Within the majority of cells, there's a nucleus. Some of our cells, like blood cells and the lens cells, the crystalline cells in our lens, don't have a nucleus. But the vast majority of cells in our body have a nucleus. And it's in that nucleus that our chromosomes sit. And it's our chromosomes that are the material that makes up our genetic information. If you unwind, so this is a chromosome that has just undergone division, so it's a pair, it's about to divide, but think of that as one chromosome. If you unwind that chromosome, it's what we call DNA, and DNA is a double helix, and so I'm sure most of you would have heard of DNA being a double helix. 1953, Nobel Prize for Crick and Watson for identifying the structure of the double helix. It's wound up in these chromosomes. If you took all the DNA from a single cell, which is something you cannot see, except unless you have a microscope, you can't see a single cell, but you unwind all of the chromosome material and it will form 2 meters long DNA. It's just amazing how that is organized into the chromosomes within a nucleus. Now, the DNA structure, it's a double helix, so there's 2 strands of it, and it's actually got a very simple, exquisite structure. There are letters that many of you are familiar with that we talk about when we talk about DNA, A, C, G, and T. And there's an absolute pairing of these different letters. An A will always pair with a T. It will never pair with a G or a C or an A. And it's that pairing, that exquisite pairing, that forms that double helix structure. Now, for some of you who might be chemists or interested in chemistry, those letters that you frequently hear when we talk about DNA, A, T, C, and G, actually stand for nucleotides. They're actual chemical entities, and it's the chemistry that causes that binding. So it's the chemical bonding between an A and a T that is critical. An A cannot bind to an A because the chemistry doesn't allow it to. So just be aware that these are, we call them A, T, C, and G, but they're chemicals that form the double helix. Some of you will also, maybe sometime during your lifetime or in certain circumstances, have had your karyotype analysed. This is the karyotype of a human female. So all the females in this room, myself and all the females, would have such a karyotype, where a cytogeneticist will do a chromosome spread of cells, and this is what they see. You see 2 copies of every single chromosome. There are 23 pairs of chromosomes, 22 autosomes, and so we have 1 copy that we inherit from our father, 1 copy that we inherit from our mother. This is basic genetics that many of you are familiar with. The females in the room have 2 X's, and the males in the room have 1 X, and they have a little Y chromosome that is different from the X chromosome, which gives us our gender. Now, the important part of this is if you then take, so just imagine that previous slide that I showed you, you take all of those chromosomes, unwind them all, it'll be a string of letters. That string of letters, and you'll notice there's only 4 letters. There's no other letter in our genome, only ever 4 letters. This particular slide shows 1,000 letters in a given sequence. Our whole genome, each and every one of us, our whole genome is 6 million times this number of letters. So you imagine that slide, multiply it 6 million times, or imagine everybody in Sydney, which has a population of some 5 million people, give those a different copy of those letters to every individual in Sydney, get them to hold it up, still need an extra 1 million people to form all the letters that form you, me, every single one of us. It's a lot of information. So just sit back and think about all those letters that form our genome. So what is the genome actually? What does it actually do? And for humans, for the human genome, which is what we're really interested in here, it's an instruction manual. So what it actually does, it provides all the instructions for making a fetus from the point of conception, of when the egg is fertilized, through the point of time where the baby is born, through the time of postnatal childhood growth, through the point of adulthood, where two individuals come together, their whole reproductive system is programmed within the genome, and starts the process and the life cycle all over again. It's all programmed in those letters. So what those letters stand for and code for exactly is all the parts that make up our cells and make up our bodies. Codes for all of that. It codes for the way, the instructions in which all those different bits are assembled into cells, into tissues, into the whole body. It codes for how all those different bits function properly. And it also functions importantly for how we can repair our cells and our tissues when there is damage from all kinds of factors. So it's what we call the instruction manual for making a human being. And you can imagine that if there's something wrong with that sequence of letters, then some of these things go wrong because the code is wrong, which causes us disability, disease, all kinds of medical problems. So what do we know about those 6 million letters? So we go back to the DNA, the double helix. We know quite a lot about parts of that genome. We know that bits of it code for genes, and genes are the double helix letters that through a messenger molecule will eventually code for 20,000 different proteins that are made in our cells. There's only 20,000 more or less genes in that genome. They are the parts of the genome that code for all the structural building blocks and all the machinery that goes on to make the cells function. But only 2% of the genome is in genes. So if we look at the genome, we now can almost accurately show where those genes sit within the genome. And let me just go back to tell you that a gene, and you'll hear this terminology, so while it's not hugely important, some of our speakers will refer to what we call an exon and an intron within a gene. So genes, it's complex, and so a gene isn't just one bit of DNA, it's one bit separated by something that doesn't code, and then there's another bit, and then there might be several bits that then eventually come together and code for a protein. So in the genome, when we look back at the letters, we see a lot of letters, then we see a region that codes for an exon, then there's this intermediate bit, then there's another exon, and then there can be like millions of letters that don't code for proteins. And there's a lot of research going on to try and figure out what all of these other letters are. And John Mattick, for example, our executive director, has been doing a lot of work on trying to identify some of the coding elements that are not proteins, but the regulatory elements that sit within these huge areas of the genome. And what you might have heard about as junk DNA, we now think that it's actually not junk DNA, but it regulates a whole lot of processes other than making proteins. We know this is quite complicated, and I'm not going to dwell on it, but we know quite a lot about genes because since the 70s, we've been able to cut bits of those sequence out. We've been able to put them into bacterial plasmids. We've been able to reproduce those bits of DNA that might be a few thousand letters long. They might be a bit longer than that, but we can put them into bacteria. We can express them. We can analyze them. We can move. We can change everything about them. And for the last 30 years, my colleagues and my own lab have spent doing this on many different genes to try and understand what they do. And the basis of a lot of the gene therapy kits that have been tried over the last few decades are really based on being able to cut and paste bits of those letters into a bacteria, put it into a mouse, put it into something else, chop it, change it, switch it around, take the bits that are interesting and do all kinds of modifications to understand what those coding genes do. But what's the rest of the genome do? Really, and how is it all regulated? How is it put together into a human being? We really don't yet understand. So we know that probably a large part of that 98% of the genome must be what makes us into a human being. If you took any of these animals that are shown here on the screen, you took their nuclei, you took their chromosomes, you took their DNA, and I put it there on the screen, it would be A's and C's and G's and T's. No other letter. So they have the same, in principle, genetic code as us, but our genetic letter, the order of the letters, the sequence of the letters, makes us into a human being, whereas it makes something else into a frog. So a real question is, what's all that about? And a lot of scientists around the world are interested in that. So, and that's what we call comparative genomics. There's also comparative human genomics, where Vanessa Hayes and the Garvin and a lot of other people are looking at human evolution from the perspective as how have those letters changed as evolution has proceeded, and what does that change mean? But anyway, we're not interested in this particular Garvin. What we're interested in is all these people that run the City to Surf, all of you in the audience, and I don't see any identical twins. Are there any identical twins in the audience? No identical twins. So all of those people, all of us, the interesting thing for us as a medical research institute is what in the genome causes us all to be different. So you look at the person next to you, you can see, you can look at their passport photograph and you recognize them. Our genome is what's made us effectively different. So let me give you some information. If you look at the person next to you and compare them to yourself, there'll be 3.5 million differences in those letters between you and them. Between me and you, there's 3.5 million differences between each of us. It's a lot of differences. So genomics, to cut to the chase, genomics is about understanding what those differences mean and what those differences in letters contribute to the variation in human phenotypes. So I'll use the term phenotypes. It means the differences in our physical attributes. Some of us are tall, some of us are short, some of us are different, look different, different colour hair, different colour altogether. So what causes all of that? Some of those differences will cause diseases, directly will cause diseases. Some of them will predispose to diseases. Some of them will affect our metabolism, our physiology, without causing disease. Others will affect the way that we respond to drugs and to medications, to caffeine, to alcohol, to all kinds of things. A large majority of them will have no significance whatsoever, we believe, at this point in time. So what we need to do in terms of studying the genome, which is genomics, is match all of those things up. So how do we do it? And first of all, before how we do it, what those differences mean, when I say 3.5 million differences, a lot of them are single letter changes. So think of 6 billion letters that make up your genome. Some of them is a single change in the letter. If it's in an exon, it's quite likely that it will cause some problem, but it's not necessary that it will cause some problem. It can also be in one of these regions that we don't understand very well, can also be some missing bits of sequence. So it could be one letter missing, 2, 3, 100, 1,000 letters missing between my genome and your genome. Could be lots of and bits of the genome that amplify and change. So there's lots of differences when we talk about those 3.5 million differences. So how do we study this? There are two main ways, and these will be described for you in our speakers. One of them is if there is an individual with a rare inherited disorder that is clearly, the clinicians will say it's clearly a genetically inherited disorder. We can take that individual and their families, do the genomic sequencing, and identify what that disorder is. at the genetic level. But a lot of the complex diseases aren't so straightforward. And so what we do is take individuals in the population that might have a similar disorder, like type 2 diabetes or schizophrenia, and see what their genome might have in common. And what we do then is match that up with members of the population who are disease-free at an elderly age, what we call 70, it's not really elderly, but that's the cutoff, of a disease-free reference group that we can use to say, to match up what the causative genes might be. It's really complicated for these kinds of disorders. It appears to be, and we'll have some talks on this, that it may be 2 or more changes that are causing a disorder. So the analysis becomes very complicated at that level. So we call these polygenic diseases. It's also very clear that the environment interacts with the genome to influence whether disease occurs. So we need to understand all of those parameters within a study. So what we now, at this point in time, what we understand about those gene phenotype associations is the tip of the iceberg. And what we need to do in order to get better understanding is research in these areas. We are at the Garvan Institute, you may be familiar with, I think you've got some material in your folders, set up the Kinghorn Centre for Clinical Genomics since 2012. And the first place in Australia and certainly the Southern Hemisphere that has now bought the machinery that enables us to get the whole genome sequence from individuals quite quickly and at a cost of, it's US $1,000, that now enables us to be able to do this kind of almost routinely, or at least for quite a number of individuals. So we can do high-performance DNA sequencing, and I think Leslie will talk a little bit more about that capability. So we need, in order to do this, we need to be able to sequence individuals. And that's for the first time, we really, for the last couple of years, have been really in a position to be able to do that. Can't be done without amazing computing power and analytics. And so we have that, that capacity building up here. In order to store your genome or my genome and be able to analyze it, we need 200 gigabytes of space on a computer. Now that's probably all the space that some of you have on any one computer. Leslie will tell you that you can store your genome on a memory stick. You can store it, but you can't analyze it, can't do anything. So it requires, to study 1,000 genomes, we need a massive amount of computer storage. We need also informaticians to be able to filter all of this stuff out. A lot of it now is just a computer process that the computing, you shove it in, bring it out, and it says out of those 3.5 million, possibly 1,000 are important. And then it's up to the clinical geneticists and clinical specialists in that field to make sense of what all that means. So it's not yet computerized. Maybe one day it will be, maybe in 20 years' time. I'm going a bit over time, but I just want to give you just a quick snapshot of when we talk about an inherited genetic disorder means that all of our cells in the body carry that. So we've inherited that from our parents. When we talk about an acquired genetic disorder like cancer, it means just that genetic change has taken place in very few cells and is amplified within the body. So that means that throughout life we have the same inherited genome, but as we age, we acquire genetic changes in those letters. And that makes a difference in how we study diseases. So the bottom line is that by linking all of those letter changes to the phenotypes and the diseases, we get a better understanding of disease biology. So in an individual, we can treat the actual cause of a disease and personalize it. We can improve genetic diagnosis, prediction of disease, and disease prevention, and I'll talk about that a little bit later. And we can develop those personalized treatments based on based on genetics. And it's this linking those genetic variants with disease and predisposition to disease, and the ability to do it, which we now call the revolution in medical research through genomics. So that's basically what we're talking about when we talk about the revolution.